Thank you for joining today's webinar, The Integration of Omics in Microbiological Risk Assessment. This webinar is sponsored by ILSI Europe. It was organized by the IAFP Microbial Modeling and Risk Analysis PDG. For best viewing of the presentation material, please click on Maximize in the upper right-hand corner of the slide window and then Restore to return to normal view. Audio is being transmitted over the computer, so please have your speakers on and volume turned up in order to best hear. A telephone connection is not available. Questions should be submitted to the presenters during the presentation via the questions section at the right of the screen. It is important to note that all opinions and statements are those of the individuals making the presentations and not necessarily the opinion or view of IAFP. This webinar is being recorded and will be available for access by IAFP members at foodprotection.org within 1 week. At this time, I will turn the webinar over to Elias Rito with ILSI Europe. Thank you, Elias.
Good morning to our participants from North America, and good afternoon for our participants in Europe. I'm here to introduce ILSI Europe, the organization that has collaborated with IAFP to make this webinar possible. So ILSI Europe fosters collaboration among scientists from industry, academia, and the public sector to provide evidence-based scientific solutions and to pave the way forward in nutrition, food safety, consumer trust, and sustainability. So as you can see on the slide, we try to build a multi-stakeholder science-based solutions to have a sustainable and healthier world. So this is done by a tripartite approach that is fundamental to the pillar of ILSI Europe. In ILSI Europe, this collaboration would not be possible without the experts, and we are divided on task forces that is specifically involved in nutrition and food safety. So we currently have 21 task forces and 45 expert groups. So these are groups within task forces that try to identify solutions in terms of food safety and nutrition. We have member companies, and we also work with academia. So the task force that has collaborated with IAFP specifically for this webinar is the Microbiological Food Safety Task Force. And the Microbiological Food Safety Task Force tries to provide guidance on microbial food safety issues to support society in implementing efficient food safety systems. So as a small or a brief background, so foodborne diseases are common, impacting costly yet preventable public health problems. So some of these factors, such as climate, global trade, the usage of new ingredients, and consumer behaviors are changing. And these changes might affect microbial populations in food. To address such complex changes, instead of addressing issues individually, a more conceptual framework detailing how to incorporate risk analyses in food safety management systems is needed. So here, the objectives and tools that we try to make in this task force is to publish guidelines and work on an agreed terminology, to understand potential detection, control, and management procedures, to review and summarize knowledge on pathogen behavior, ecology, and to assess the risk to consumers, as well as to develop these tools to manage safety hazards and risk in food production systems. We engage with our stakeholders through various ways, such as peer-reviewed publications from the expert groups, webinars, workshops, and we are also involved or has been involved in European projects. After the presentation, we will be showing to you what will be our upcoming activities. And I would now want to give the floor to our moderator, Dr. Alejandro Mesquita, to give more information about this webinar.
So thank you, Elias. Yes, my name is Alejandro Mesquita. I work for Unilever based in the United Kingdom. And it's a pleasure today to be the moderator for this webinar. A little bit of the background of the webinar. This about 3 years ago, linked to the IFP European meeting in Athens, Greece. There was a workshop that brought together academics, experts from industry, and experts from different regulatory agencies to discuss the use of omics data in the context of microbiological risk assessment. The output of that were 4 papers that have finally been published as open access in a special issue of the International Journal of Food Microbiology. So if you want to know more about the full content of the output of this workshop, you can go to volume 287 of the journal, and the link is on the screen. And today is a pleasure for me to introduce 3 speakers that were part of the workshop, and they will be going through 3 of the papers that came out of the workshop. So we start with Dr. Heidi Den Vesten from Wageningen University, who will be talking about the use of omics data in exposure assessment. Then Dr. Anna-Marie Pilat from Unilever R&D in the Netherlands, talking about the also the use of omics data on hazard characterization. And we close with Professor Luca Coccolin from the University of Turin, then bringing us into the future, looking at the integration of multiple omics data types and that need for integration of data. So with that, I hand over now to Heidi for the first presentation.
So it's my pleasure to kick off, and I will start with the exposure assessment topic of today. Because exposure assessment plays a central role in quantitative microbial risk assessment, and today I'm going to talk about the potential of omics for exposure assessment. But the topic I will address today is a joint effort of a whole team of people, and I especially want to mention Jean-Marie Membray, who shared our writing group. So first, an introduction to quantitative microbial risk assessment. According to the Codex Alimentarius and QMRA, consists of 4 steps. In the first step, the hazard identification, the question is answered which pathogens are associated with the product and which can cause a potential adverse health effect. And then the second step, the exposure assessment, quantifies the kinetics of the pathogen along the food production chain in order to quantify the dose at consumption. And then the hazard characterization focus on the evaluation of the chance to get sick and the severity as function of the ingested dose. And then finally, the risk characterization integrates the information of the hazard characterization and the exposure assessment step. Because when a dose is known and also the dose response correlation, then the chance to get ill and the severity can be estimated. And when a dose is estimated, including variability encountered in the chain, then also the chance to get sick can be expressed with its corresponding variability. So omic tools have been developed last decennia and paved the way for an extra dimension in QMRA. And omic tools are used at different levels in the cells, from DNA analysis to metabolite analysis, and can help us to answer different questions. The genome sequencing of strains or microbial consortia give insight in which microbes are present and what these microbes could do. And then the genome-wide transcriptome analysis shed light on what appears to happen. The proteomics target the protein levels and give insight in the proteins that makes it happen. And then the metabolite analysis help us to understand what kinds of molecules are produced, or in other words, what has happened and is happening. So the omic tools allows us to gain insight into the responses of cells to changes in the environment and their potential to cause disease. And omics can be employed from single strains to whole consortia of strains. And when the whole consortium is addressed, this is called meta-omic analysis. And Luca will talk about it later today. So back to the exposure assessment. The question which is answered in the exposure assessment is how will my troublemakers behave? And for that, the target, the levels of the pathogen and the kinetics along the food chain from farm to fork have to be quantified. So we need quantitative numbers for possible growth, inactivation, survival, and also contamination. But one of the main challenges in exposure assessments is that we know that microbes are not all the same, and we have to take into account microbial variability in our predictions. And also foods, they are complex ecosystems, and the pathogens are part of these complex ecosystems. And we think that omics can help us to improve our predictions of microbial variability and to better understand the dynamics of pathogens in complex food ecosystems. And today I will share with you our thoughts how omics could make a difference, but also the challenges we are facing with the omics. So we all know that microbes are not all the same, and because we do not know which strains we will have in our product, we have to address strain variability. And how can omics then help to better characterize strain variability? A good example of strain variability is the diversity we have seen for Bacillus subtilis strains. Together with NISO food research, we characterize the heat resistance of spores of 20 different Bacillus subtilis strains. And this is what you can see in the left figure, the log D value as function of temperature. And when we then calculated the average Z value of the spores based on all 20 strains, we got a very high Z value of 51 degrees Celsius. When we then had a closer look at the data of these 20 strains, we could actually see two different groups. A group of 11 strains that produce spores that were extremely heat resistant, and 8 strains that produce spores that were less heat resistant. And grouping of the strains into these two groups also resulted in a more realistic Z value of the two groups, as you can see. And because the heat resistance was clearly different between the two groups, our NISO colleagues investigated whether this could be supported by genetic differences between the two groups. And for that, they sequenced all these 20 strains and followed a gene trait matching procedure to see whether the differences in heat resistance could be explained by a genetic biomarker. And indeed, they found that a specific transposon element was only present in the most heat-resistant group. And when this element was deleted in one of the heat-resistant strains, indeed this strain became part of the heat-sensitive group. Also, when this element was integrated in a sensitive strain, this strain jumped to the group of the heat-resistant strain. So this is a strong indication that this genetic element can function as heat resistance biomarker. And promising biomarkers can help to make subgroups of strains and thereby reducing the variability within a group. This allows to fine-tune exposure assessment by doing the exposure assessment for the different subgroups, taking into account the phenotypes of the different subgroups. But then we need quantitative data, not only for heat resistance, but also for growth. And how could this work? I would like to explain this by using Bacillus cereus as an example. The Bacillus cereus group is a large and very diverse group, and based on genetic signatures like cold shock proteins, 7 groups could be assigned. So based on genetic biomarkers, the group division is made. To fine-tune an exposure assessment, then the data is needed for all these 7 groups. And the French group of Cunebritaire determined the growth ranges of these 7 subgroups. And as you can see in the table, the most psychotolerant group is group 6. Next to growth, for exposure assessment, you also need info on heat resistance. And this was determined by the group of Chris Michiels. As you can see, the heat resistance of the 7 different groups differed largely. and therefore it's very difficult to compare the strains by comparing the D values at only one temperature. But they used a very nice method to compare the strains, namely to determine the temperature where the log D value equaled to 0.8. So then strains are compared with respect to the temperature at the reference log D of 0.8. You clearly see that the differences between the groups result in differences how the strains can, for example, survive in heat treatment at 90 degrees Celsius for 10 minutes. A strain belonging to group 3 was not inactivated within this time period of 10 minutes, while a strain belonging to the blue group, more to the left, was reduced with 2.8 log during these 10 minutes of heat treatment. So it's clear that there are clear differences in heat resistance of the different groups. And they also found a positive correlation between growth and heat resistance, because the psychotolerant strains were less heat resistant compared to the strains that had a higher growth limit. So based on genomic investigation, we could make subgroups. And when we then collect phenotypes of these different subgroups, the exposure assessment can be done for the different subgroups. And this gives then more precision than when you take the whole group as a whole. So far I have talked about how omics can help to understand how we can fine-tune exposure assessment based on mechanistic insight of subgroups. But the second domain where omics has the potential to make a difference is to better understand food ecosystem dynamics. Because in the food, pathogens are not alone, and microbial communities affect the dynamics of pathogen. And how can then omics help? In this case, metagenomics can help to understand the dynamics. We can use metagenomics to characterize the communities, to understand which species are present in food, but also in our food environment. And this can also help to elucidate transmission routes of our pathogens. And there is a clear interest to gain more insight in the ecology of the food and in the factory, and also to have a quantitative estimate of the various species and also the changes of the species in time. But the first step to answer this difficult question is actually database building based on our food samples. We need data, we need metadata on the food characteristics, like for example pH and water activity, but also extrinsic factors like temperature. And then at the same time, samples are taken to determine the concentration of contaminants with plate counting. And then in parallel, metagenomics analysis are done to determine the composition of the community. And especially this combination of plate counting and metagenomics makes it very strong, because then the concentration of the different species can be determined at the same time, but also the corresponding changes over time as function of the food characteristics. And then this allows to model the dominant species linked to the food characteristics. And then this information is stored in databases. And then the next step is the prediction. So in the next step, we want to use the information to predict the kinetics of the species in new samples. So when a new sample is taken and a microbial composition is determined in that sample, then a database and the models can be used to predict the population dynamics over time based on the food characteristics. But the prevalence of food of the prevalence of pathogens in food is low. And of course, this makes it very challenging to also collect metagenomic data, including the pathogen. And Luca will also address this in his talk. But this approach is not only relevant for food safety, but also for food quality, and thereby more focusing on the dynamics of dominant spoilage organisms in the food. So, in conclusion, how do we see the potential of omics in exposure assessment? First of all, omics helps to understand the biology, the composition of the food ecosystems, and the microbial dynamics of these systems. And next, biomarkers can help to better understand mechanisms for strain variability. And when these biomarkers are validated, they can also be used to predict strain variability. And when we are able to better characterize pathogens of interest, we can subgroup them. But then we need also data on relevant phenotypes of the subgroups, like, for example, data on growth, inactivation, and prevalences. And this is a big effort. But if you have this data, you can then fine-tune exposure assessments by reducing uncertainty. And with my last slide, I would like to conclude with my acknowledgement of all the members of our writing group. And again, a special thanks to Jean-Marie, the chair of our group. And also thanks to all of you.
Okay, thank you, Heidi, for introducing on the first step in one of the first steps in quantitative microbial risk assessment, and that is exposure assessment. And once you are exposed to a certain organism, you digest it, and of course you then get to deal with some dose response, which we call hazard characterization in risk assessment. So in this part, I will step back a little bit and introduce you to hazard characterization in quantitative microbial risk assessment. So as from the presentation of Heidi, she already mentioned that you could have some variability that you could include, but suppose now first that the dose that comes out of our exposure assessment is, for example, 3-log Salmonella typhimurium that you found in a sample on your plate that you're about to consume. Then using a dose response model will give you the possibility to calculate the probability of becoming ill from consuming this specific dose. And of course, there is several or multiple number of models, and the most simple one is an exponential model, and which in the lower dose range can be described actually by this following simple linear equation, which has here 3 parameters. It's alpha, beta, and D is the dose that you are exposed to. This alpha and beta could of course be point estimates or average values that could describe your data. But as you see here in the picture of Peter Teunis et al., who published it, is that alpha and beta can also include some biological variability. For example, based on the susceptibility of your cases of illness. And so biological variability can be included into these type of models. Another way to get compared to epidemiological data, you could also get those response relations from doing in vitro experiments. So, for example, doing experiments that represent your gastrointestinal tract, and repeating those kind of experiments means that you don't always get the same results when doing totally what you're supposed to have a totally similar experiment. If you would repeat that twice, then you won't get to the same output, and that is because of experimental uncertainty. And you could both include biological variability and experimental uncertainty into your models. And I think it's important to do that, because if you include uncertainty, you are able to distinguish that from true biological variability, and what you are looking for in your dose as the output of your dose response models. So if you do this, then you can come up with a response that is the number of ill people, and I have an example here on how we did a dose response model for portions of mixed salad that are eaten raw. So you can buy in the shops different mixes, which I call here mix 1 to 16, so 16 different types of mixes. And you can imagine that the produce that is being used to get into these mixes can have different concentrations and prevalences of certain pathogens. And that is what we analyzed. So we analyzed throughout the whole exposure system. We analyzed bags of mixed salads, both its produce and the products, on pathogens that were in there. And we found the results that in those mixes, we found in 12 out of the 16 mixes Salmonella typhimurium, however, with a different prevalence and with a different concentration. That means that if you want to calculate the probability of becoming ill from eating a portion of this type of mixed salad, differs among those mixed salads. Also, Campylobacter was found, and in one we found E. coli. Salmonella montevideo was found in one of the products bags. So this was all found in produce, and this was found in a product. And what you can do then with a QM array is calculate, well, of course, the mean number of ill cases per year in the Netherlands from the consumption of a portion of mixed salad. You can calculate confidence intervals or median values, and you can already see here that if you would like to include even more underlying data than only on strain level, what is happening here in these different bags, that this can gradually become a very extensive study to do. And so that is what I would like to represent in this slide. If I'm looking at dose response alone, the input would be, say, in dose response survival in your gastrointestinal tract. You could also look at growth rates or other things that are related to dose response, and that means that basically you would have phenotypic information in something in the 1, 2, 3, or 5 different characteristics. And from that, what we normally do from that, we calculate a dose response having as an output number of ill cases that has one dimension. What will happen if we have, instead of only this type of information, also genotypic information? You can imagine that this then can become quite difficult, and that the number of hazards can increase very much depending on how far you go with having genotypic information on the strains that are that you found during your exposure assessment. And the question is then, of course, the challenge is how do we map this? How do we map this genotypic information to phenotypic information, and then to one dimension, and that is the number of ill people. There is a number of challenges, and we should remember that risk assessment is, of course, intended to support decision making, and this decision making can become very difficult if we get these kind of new data sets that influence risk assessment directly. there is many questions and even more answers. And I named a few here. In the paper it says something like, if you would then find a new genotype, would that also identify a new hazard? How would that change then or impact for policymakers? Another question is, how does the presence or the absence of a virulence genes characterize a hazard or a non-hazard? If you find a virulence gene, does that mean that you have found another hazard or not? And when it comes to all kinds of transcriptomics data or proteomics data, these are most of the times characterized with differential expression. That is how the statistics behind these proteomics works. But if you find a differential expression, does that mean you have a different type of risk? And not even to mention on how do we use then molecular data analysis for probabilistic calculations that are at the moment used in QMRA. So let's see how this would work if we look from a governmental side. If we look here at ESTEC, you can easily see in table 2 that if you have sequenced of a more and more stricter definition of your pathogenic potential, then within regulation, you can come up with a lot new era of rules. And I'm not going to read that out loud here, but you can see already that in the Dutch guideline of 2014 for ESTEC, the differentiation of serotypes and the genes that are involved with that makes that regulation can become very elaborated in the end. And this is only for ESTEC. So if we look at challenges from an industrial perspective, well, in a factory, there is a quality manager who needs to make decisions. And the question is, if you have incorporation of omics data that for a quality manager often has little biological meaning, if you put that into risk models, this will require input from the risk manager and will make the decisions making processes even more complex. So there is a concern here around the lack of standardization and reproducibility of current methods to actually affect decision making of quality managers in a factory. On the other hand, in addition to this, there is a lack of relevant data sets. As Heidi talked about before, it's a lot of literature is available on pathogenic organisms, whereas in a factory we need to be aware of spoiled organisms also, and that is something that is not often being addressed during research projects. So in the end, our paper describes, if we take all of this into account, what are then the potentials of NGomics? And I think that the potentials here are very much on hazard identification. You can provide greater details on pathogens when you look at omics data. It can say something about variability in pathogenicity, in virulence. It can reveal things on stressor responses like Heidi already introduced, and it can say something about the interaction with other systems, both for humans and microbes. If omics can be used to identify biomarkers, then that is also a very big area of interest, because if you identify a biomarker, it means that you don't need to do all kinds of challenge testings anymore, of other kinds of testing in order to find some phenotype information. But the question is then, of course, how do we translate the biomarkers from human and cell culture and for example, animal models responses into those response models, because that is what we are addressing currently here. As it comes to the biomarkers, the paper that was written gives a nice overview of the kind of omics methods that are out there and that have been applied, the type of biomarker, an example that goes with it, the type of response, something about reproducibility, and of course the correct reference that goes with that. And I think that this review gives a hint about what type of biomarkers and how you can come up with biomarkers that would represent the strains that are important for your dose-response relationship. So, oops. In addition to only finding the ways of finding biomarkers can also be done through network analysis. And the paper that we have written says something about how a regulatory network of 10 Salmonella strains was constructed based on 233 genes that are associated to virulence. Which have been described in a virulence factor database that is present on the internet. And through this kind of analysis, you can find, for example, the main regulators in this data for those 10 salmonellas, which are presented here as genes. So if we think about biomarkers and those response, how would we go about that? So how would we correlate biomarkers to responses and illness conditions? Questions come up like, how do we quantify these correlations and how will it impact those response? As a basis, you could, for example, have a look at the WHO FAO report of 2002, where a lot of work has been done on getting a dose-response relationship for Salmonella. And what these lines here indicate is the thick line is the expected value for the probability of illness, and the dashed lines here around it give the 95% confidence interval about this illness, and the thin dotted lines give the upper and the lower bounds. And you can see here that already uncertainty is very large at the low infectious dose, which can be which is shown here on the right hand side. So the question is, how can we include biomarkers in getting maybe the uncertainty of this kind of dose response relationships to become smaller? And for that, we think that when it comes to omics and dose response relationships and the ways forward is that the type of data, if it is biomarkers or other genetic types of or omics type of data in microbiology, so on the strain itself, should be linked with other data in order to get a representative output. So, for example, combining it with epidemiological and process data. And in that way, you end up with including systems biology to come to an expression of illness during your QMRA, because, well, the presence of a biomarker, may it a gene, a metabolome, a protein, may by itself not always be a very good predictor, since the expression is influenced by a large variety of biological factors, and of course biomarkers may be dependent. So, in conclusion, I think that what we see around us is that omics is already changing the food industry. In the next few years, it's going to impact industry's ingredients and products specifications, surveillance programs, and detection methodologies. It may also increase the chances for companies distributing products across different regulatory environments, because of course there might be, depending on where you distribute your products to, there might be implemented different rules, for example, comparing the US already with how things are going in Europe. Though I think that if we collaborate effectively with academics, regulators, people from government agencies and industry, the impact of omics on microbial risk assessment will improve the quality and accuracy of our hazard characterizations. So, thank you. Let's move on to the next presenter.
So, good morning and good afternoon, everybody. My name is Luca Cocolin. I'm from the University of Turin, and I'm going now to try to take you in a futuristic view of how the meta-omics can be used for the improvement of microbiological risk assessment. Just a short message from the moderator. If you have questions, please just type it and do not wait for the end of the presentation, because the moderator can already see the questions and can organize the questions for the end of my presentation, when the discussion session is going to be open. So, before getting into the actual topic of my talk, I would like first to underline how the way we are looking at ecology in foods, and when we're talking about ecology in foods, we also take into consideration, of course, foodborne pathogens, has been changed throughout the years. And especially in the last 10 years, when culture-independent methods started to be applied, we realized that we cannot anymore think about single species or single populations, but we have to talk about consortia. And we need to take into consideration this aspect, because consortia are complex ensembles of microorganisms that they can interact, they can communicate, and of course they can impact their behavior, and as a consequence what they do in food and what they do into a human body, for instance. And so if we simply take into consideration the technology advancements throughout time, as you can see, we started with the traditional methods based on plating, counting, and isolation. When culture-independent methods started to be applied, we were able to fingerprint the microorganisms' presence in the complex ecosystems like foods, for instance, with fingerprinting-based ecology methods like DGG. And then the technology, when the technology advanced, we switched to sequence-based ecology studies like metagenetics or metataxonomics. This is a term that I'm going to use it later. And at last, what we have experienced recently is the actual use of functional-based ecology studies like metagenomics or metatranscriptomics, where we can understand what microorganisms actually can do within an ecosystem. But of course, the challenge is not over. We have to try to take advantage of the information that we have collected or we are collecting through functional-based ecology to go back in the laboratories, to go back to plates in order to be able to cultivate the microorganisms that are expressing the most interesting features that we can exploit at different levels. And of course, now this part of the studies are started to take action. We are talking a lot in the last couple of years about culturomics, and I think that this is going to be something that we will really need to look very carefully, because we need to somehow take advantage of the big body of knowledge that we have collected and we are still collecting through the functional-based ecology studies. In this slide here, you can see a very comprehensive figure where the opportunities that, in particular, next generation sequencing, and we're talking about specifically here next generation sequences, can give when we want to look into the ecology of foods, clinical samples, animal samples, or environmental samples. As you can see, we have, first of all, we have two different possibilities, because we have the choice of working on the DNA or working on the RNA. We need, of course, to recognize that those molecular targets have different meanings inside of a cell. So the DNA is something that is stable and is actually delivering the message, the message for, you know, whatever is necessary to the cell for the functioning. And on the other hand, we have the RNA that is something that is very labile, is not stable, and so this is really a molecule that can give us an information on the activity that the microorganisms can have in a specific ecosystem. We can use amplification, and so we go into metataxonomics. We can have amplification of the DNA or amplification of the DNA of the RNA, depending on the study that we want to make. But again, those metataxonomics will just give us an idea of the ecology. So when we talk about metataxonomics, we know that we have a phylogenetic view of what is happening inside of our ecosystem. Of course, when we take into consideration the lower part of the graph, meaning metagenomics and metatranscriptomics, at this stage, what we do is to really look into the whole DNA or whole RNA that is present inside of an ecosystem. This to me is actually the big excitement in doing this kind of studies, because looking at metagenomic libraries, for instance, we can reconstruct much more precise the taxonomy of our ecosystem. We can also look in the function, because of course the number of the genes that are going to be present, they will be responsible for a certain function if these genes are going to be expressed. And most probably what is going to give us the next let's say the next possibility to really look into the intraspecies diversity within our ecosystems with a culture independent method is going to be the whole genome reconstruction. So from the metagenomic libraries, we can reconstruct the genomes of our microorganisms that will somehow highlight the number of the strains that we have inside of our ecosystem. And when we look into the RNA, at the level of the RNA, as you can see, it is going to be important to understand what are going to be the transcribed metabolic pathways, and this is going to be extremely important if we take into consideration, for instance, spoilage or also fermentations, because microorganisms with their metabolic pathways, they will really influence what is happening in our ecosystem. Said that, I just want to take you in some examples where we have used those methodologies for understanding a little bit better what is happening in different complex ecosystems that are responsible for two types of food fermentations. The first one is the one that is taking place during cocoa beans production. Here you have a study that we have published at the end of last year, looking at the cocoa beans, and in particular what we have done was to compare the yeasts and bacteria ecology in two different methodologies that are, one very traditional, that is the heap fermentation, and the other one that is a rather innovative way of fermenting cocoa beans, the one that is in the box. And what is highlighted in these two tables, the data are expressed as relative abundance, so there are percentages of relative abundance. You can see that while the bacterial population is rather stable, and so you have a certain number of major players that are always constant independently from the type of fermentation or the technology of the fermentation, that are the ones that are underlined in red. On the other side, on your left side, you can see that the yeast ecology is highly dependent on the technology that is used. So there are different species of yeasts that are responsible for the fermentation depending on the type of technology that you're using. And so this is already a very first indication that we need to take into consideration the technology in order to drive some specific fermentations and to promote some species instead of the other ones. And if we analyze the data that we obtained by metataxonomics and we correlate the presence of bacteria and the yeasts, you can see that the fermentations that have been conducted in the box and the fermentations that have been conducted in the heap are rather different. And so there are positive and negative correlations that we can understand based on the type of technology. And even more, if we add in the analysis the volatilome that we have determined in these two types of fermentations, you can really understand how the microbial actors that are present during the fermentation can impact again on a positive or negative, in a positive or negative way towards the production of specific volatiles. So to summarize, and of course you can get more detailed information reading the paper, you can understand from ecology to volatilome how the microorganisms are interacting, and you can somehow use the technology or change the technology that you're using for the fermentation in order to promote or to limit the development of certain species, and as a consequence, the development of certain volatiles. The next example that I would like to explain, or I want to report to you, is the experience that we had with metagenomics. This is a real metagenomic study working on fermented sausages. What we have done here is very simply two types of fermentation, one that is spontaneous, the one that is coded S, and the one that is inoculated, the one that is coded Y. I mean, you can see that in the inoculated fermentation, you have the predominance of two species, that is the Lactobacillus sakei and the Staphylococcus xylosus, that were the two species that were present in the starter culture, while on the other fermentation, in the spontaneous fermentation, you have a higher diversity of microorganisms, and this is absolutely confirming the current knowledge that we have on spontaneous fermentation. But again, Lactobacillus sakei and Lactobacillus curvatus were the two main actors of this fermentation. We have done some metagenomics out of these samples, and we have reconstructed this network where we have compared the metabolic activities clustered in different types of metabolism, like the proteins, the carbohydrate, the amino acids, and what you can see, and the other ones that you see in the slide. And we compared them with the points of the fermentation. And as you can see, the inoculated fermentation at 3 days, it seems to express at max the activities that are responsible for protein metabolism, cell wall synthesis, carbohydrate fermentations, and a lot of other ones. So the again, time 3 for the inoculated fermentation is the time where most of these activities are very well present, while the spontaneous fermentations, you can see that are a little bit more on the edge, and so meaning that the activities are a little bit more slow. But at this point, I would like to come back to the main topic of this webinar. We talked about omics, we talked about risk assessment, and so I would like just to give you a couple of insights on how, in our opinion, the omics can, and the integration of the omics can really help on the risk assessment. Here is a study that we have conducted recently on the ozone treatment of surfaces in a meat processing plant. You can see that we have used different quantities and different time of ozone in a gaseous form. And in the figure, you have the presence, the relative abundance of specific populations that have been determined by metataxonomics before and after the treatment. And it is clearly shown how there are some populations like the Pseudomonas that are very, very sensitive, and so they do not show up after the treatment, while there are other ones like the Staphylococcus that still are present after the treatment. And again, looking at the data, we can construct networks in which we can correlate. In red, we have negative correlations, and in green, we have the positive correlations on the ecology of the microorganisms that are present inside of our environments before and after the treatment. And so we can look at biomarkers, in this case as species present inside of the environment, in order to understand a little bit how the wall ecology is moved. And for the pathogens monitoring, already it has been mentioned before that this is going to be very challenging because the pathogens are present in very low number. However, when we look into metagenomic libraries for Listeria monocytogenes sequencings, for instance, in the previous studies that I showed you about metagenomics of the fermented sausages, we were able to find some sequences in the fermented sausage that was not inoculated with starter cultures. Of course, there is a huge difference in the number of the reads that we have for common genes, let's say, in the wall ecosystem and the ones that are related to Listeria. And so you can see that we have a max of more than 60,000 for the wall ecology, and then we go to less than 1,000 for Listeria. However, we were able to recover it, and you can see that most of them are always related to genetic information, metabolism, and also environmental information processing. And in the heat map that you see at your right, we have, these are brand new data that we have obtained, in which, again, we were able to find some Listeria monocytogenes sequences. However, they were all related to housekeeping genes or functions that are related to, let's say, life cycle of the microorganism in specific systems. Looking at specific virulence factors, here are the results. So you can see that there is a very low number of virulence genes that are present in the libraries. If you ask me what is the meaning of having five sequences that are matching with invasion-associated secreted endopeptides, I'm sorry, but I cannot answer you. I don't know yet. So going back to the conclusions, I mean, this is just a part of the text that I cut and paste from the paper that we have published. In my opinion, is very, very important that we are approaching food ecosystems with different methodologies. Here you have some metagenomics, metatranscriptomics, metaproteomics, and metametabolomics in order to have an holistic representation of the microorganisms that are present, how they behave, and how they can interact. Of course, this is not simple. You have already heard from the previous speakers that there is a need, or there is a challenge but I believe that at the moment we will really look into the challenges, and we're going to be facing the challenges probably with the integration of different knowledge, and with different expertise we will be able to tackle it. Thank you.
Can you hear me? Yep. Okay, thank you to all the presenters. I think the three presentations were really good, and they cover, give you a flavor of what the special issue of the of this workshop includes in the 4 papers that were published. I recognize we are right at the end of the hour, but I think it would be unfair not to pick up some of the questions that have come in. So if you need to disconnect, if you need to rush to another commitment or meeting or whatever, thank you for joining. I will just pick maybe 3 or 4 questions that have come in the past 10-15 minutes, and then we'll stay around maybe for the next 5 to 10 minutes, if that's okay with everyone. I think the webinar is still open by ILSI and IAFP, so we can pick up some of the questions, okay? So the questions have not come specifically to a given speaker, so some of them are fairly open. Some others, I think I can guess probably to which speaker is directed. Let's start with the first question. I think this is open to the three speakers. The question is: do you suggest any free web-based tools to predict antibiotic-resistant genes, virulence factors, or any other food safety concerns from metagenomic data? So it's open to the 3 speakers.
Well, I can start probably, so that I'm breaking the ice somehow. I think that nowadays there are very good tools that are, again, free to access. However, it is very important to understand that those are going to be useful if you have single isolates. So in order to understand if, for instance, strains that you're isolating from your environment can be harmful or can have some features that are rather important, I would say yes, of course, this is going to be very interesting. Probably, I'm not sure if those tools can be run also on metagenomic libraries, meaning that you are feeding the system with metagenomic data. Of course, in that case, you will get a level of information that is a little bit more complex to interpret, because it's going to give you a kind of hint about the presence inside of your ecosystem of potential harmful microorganisms that you will not know which probably they are. I'm not sure how much conserved are the sequences of antibiotic resistance, for instance. In some cases, probably you will be able to understand if the antibiotic resistance comes from a Listeria monocytogenes, from a E. coli. In the other case, probably you will not be able to do it.
Thank you, Luca. And anything to add from Heidi or Anna-Marie? Okay, I take the silence as no. Right. The next question I'll pick in the interest of time, relevant to the content of the webinar, is I think I don't understand fully how it's written, but I think the question is: how do you take results from 5 virulence genes of 233 regulated salmonellosis, referring to the paper from Haddad 2018, and 13 virulence gene regulating listeriosis from a paper of Newgate 2018, to genus or species level microbial standards in food. So I think it's probably more to Anna-Marie, the question on how do you translate this sort of data from a limited set of genes into something that can inform standards.
So yeah, well, that is of course the complication about this. I think that the most important thing here is to link these kind of outputs from molecular methods to another type of information, and that is either phenotypic characteristics that can be from epidemiological viewpoint, or from doing other types of phenotypic data, and that combination might give you some indication about the hazard of one strain compared to another, and then you might maybe come up with certain groups in regulation that you can give priority to when it comes to, for example, intervention studies to prevent the translation of these type of strains through the whole farm to fork chain. And if you could prioritize on these, that would give you some idea on where to put your money in, for example, when you're a regulation body.
Thank you, Ana Marie. I'll pick one that is probably more relevant to Heidi, so we also hear her voice in the questions and answers. So there's a question here about, it's a long question. I'll try to sort of rephrase it in a way that it makes sense. So it's how can we basically move from traditional approaches to characterize biological variability of phenotypic responses to use omics approaches in that characterization of the biological variability?
Yeah, I think the link between like collecting phenotypic data should always be made with when you also look into, for example, genome information given by sequences of strains. This link is needed to understand like which kind of genes might be predicting specific phenotypes. But this validation of potential biomarkers, this is the hard work to be done, and also needs to be validated. And that's, I think, one of the examples I try to share today is about like inserting a gene or deleting a gene, give some evidence that it might be a potential biomarker for a phenotype you are looking at. But this validation is crucial to use a biomarker in a later stage, also for predicting phenotypes of pathogens.
Thank you. And I think in the interest of time, I'll just pick one more question, and apologies to some of the other people that submitted questions in the past couple of minutes, but I don't want to take far too long of your time. For those of you who are going to the IFP meeting in Europe, I think all the speakers, or at least most of them, will be there. So over a cup of coffee or a cup of tea, you can also grab them for questions in a few weeks in Nantes. This question, I guess, to also hear from Luca on the topic of metagenomics. So how can we translate the biodiversity shown by metagenomics into population density? That means log CFU per gram for each group.
This is going to be one of the next challenges, because I totally agree that one of the limitations that we have right now is as if we are speaking two different languages, in the sense that as microbiologists we have always been used to rationalize in terms of UFCs. And so we know, when you have 10 to the 8, that is a high population, and we know that it's 10 to the 2nd is a low population. However, when we are talking about relative abundance, and especially when we talk about metataxonomics, that we have an amplification step, where we really do not have any idea what is happening. A little bit better probably when we go to metagenomics, because with metagenomics we don't have any kind of let's say artifact that is created during the amplification. So in that case, probably the situation is a little bit more real by the ecological point of view. However, we really need to have more studies related to correlation of CFUs with the sequences. I know that there have been some projects already carried out related to that. They have created some standards and some, let's say, scales that they can be used. But I totally agree that we need to invest more time and more money in order to be able to really correlate what, let's say, a certain number of copies or a certain number of reads in terms of population density.
Thank you, Luca. So it's 5:40 p.m. in the UK GMT. So I think probably this is a reasonable time to close the webinar. Apologies, we went a little bit over time, but I was ambitious to include 3 speakers in a single webinar. But thank you to Heidi, thank you to Anna Maria and Luca for great presentations, and also thank you to ILSI Europe and IAFP for facilitating this way to disseminate knowledge that hopefully is of interest to the community of microbiologists and scientists working in public health and in microbiological risk assessment. So we can close the webinar with this. Thank you very much.
And we have a quick message from ILSI Europe.
Yes, apologies. Elias, over to you.
Thank you. So as this has been organized by IAFP and ILSI Europe, this is one of the activities that we conduct in order to disseminate the results of the task force and the expert groups. Our next upcoming activity of the Microbiological Food Safety Task Force will be a roundtable discussion at the IAFP European Symposium on Food Safety, which will be announced. And this roundtable will discuss the current state of the science on epidemiology, public health burden, and risk assessment for viruses and food processing environments. We'll have a roster of speakers coming from academia and as well as the industry. If you have further questions, more information can be found at the ILSI Europe website. And we will also have a booth there. So if you're going to be announced, please drop by our roundtable discussion. One last point of information, which has already been mentioned earlier, is that this webinar recording will be available at the IAFP and the ILSI Europe website, and we will be posting it soon. Thank you, everyone, for your time.
And that will conclude today's webinar. We'd like to thank our speakers and our moderator in ILSI Europe. A recording of today's webinar will be available on the IAFP website for members within 1 week. Thank you for joining us. Have a great day.